All right, welcome back to The Explainer. Today, we're jumping straight into one of the absolute most fascinating mysteries in modern medicine, cancer immunotherapy. We're looking at this incredible paradigm shift, where instead of just poisoning a tumor with chemotherapy, we're actually recruiting the body's own immune system to do the fighting for us. It's a brilliant concept, right? But as you'll see, it is deeply, deeply complex. So get comfortable, and let's decode the science of precision oncology together.
Okay, let's dive into this. Why do miracle drugs cure one person but fail another? Honestly, this is the central question keeping oncologists awake at night. We have these revolutionary therapies that can completely eradicate late-stage tumors in patient A, yet patient B, who on paper has the exact same type of cancer, sees absolutely zero benefit. Solving this biological puzzle, it's the ultimate frontier in medicine right now.
To get to the bottom of this, here is our roadmap. We'll start with the immunotherapy breakthrough, then cover tumor resistance mechanisms, hunting for predictive biomarkers, the challenges in biomarker testing, the 3PM framework, and finally, the future of precision oncology.
Let's start with Section 1, the immunotherapy breakthrough, and taking the brakes off.
So we're talking about immune checkpoint inhibitors, or ICIs. Your immune system naturally has these checkpoints. You can think of them as safety brakes that are there to stop your immune cells from accidentally attacking your own healthy tissue. But cancer cells are notoriously sneaky. They actually hijack these checkpoints to hide. ICIs target specific pathways, things like PD-1, PD-L1, and CTLA-4. By blocking these pathways, ICIs basically release the brakes, reactivating your immune system's natural ability to hunt down and destroy those cancer cells.
And this brilliantly illustrates exactly how that plays out on a cellular level. Normally, a tumor cell uses its PD-L1 protein to bind to a T cell's PD-1 receptor. It acts as a brake. But when immune checkpoint inhibitors enter the picture, they jump in, block that connection, and essentially cut the brake line. This leaves your body's soldier, the T cell, fully activated and ready to attack the tumor.
But here is the massive roadblock we're facing. The response rates are incredibly variable. If you look across different types of cancer, these treatments might see a response rate of just under 20% in bladder cancer monotherapy, but then that number jumps to over 39% when you use combination therapies in melanoma. That gap, that represents millions of patients. We have the weapon, but we just don't always know exactly when and where it's going to work.
Which brings us to Section 2, tumor resistance mechanisms, how the tumors fight back.
Now, what's really interesting about this slide is how it separates the tumor's counterattack into two distinct strategies. You have intrinsic resistance, meaning the tumor is practically invisible right from day one. It just completely lacks the markers for the immune system to even notice it. And then you have acquired resistance. This is when the drug actually works initially, but the tumor mutates over time, creating a hostile environment that exhausts the T cells until they just give up.
Let's quickly break down the biology of how tumors actually pull this off. First, defective antigen presentation. They actively hide by turning off the molecular red flags that tell T cells they're dangerous. Second, if we block one checkpoint, like PD-1, the tumor just upregulates a different checkpoint, like TIM3 or VISTA, forcing a brand new brake on the immune system. Third, they cause T-cell exhaustion by constantly overstimulating the immune cells until they literally just wear out. And finally, metabolic reprogramming. The tumor gobbles up all the glucose in the area and pumps out toxic lactate, completely starving the immune cells.
All of this intense biological warfare goes down in a very specific location called the tumor microenvironment, or TME. You really can't just think of a tumor as a lump of cancer cells. It's an incredibly complex, microscopic ecosystem, and it is a decidedly hostile battlefield for our immune system.
Right in the middle of this landscape is the central tumor mass. But the perimeter is where things get wild. The tumor has essentially recruited its own private security force. You have regulatory T cells, or TREGs, releasing immunosuppressive cytokines. You have myeloid-derived suppressor cells, or MDSCs, actively stopping T cell activation. They form this intense defensive shield around the cancer. If an immunotherapy drug is going to work, it has to navigate this dense, suppressive network of cells.
Moving to Section 3, hunting for predictive biomarkers, the molecular fingerprints.
Currently, doctors rely heavily on two major biomarkers to navigate this battlefield:
The first is PD-L1 expression. This involves taking a tissue sample and literally counting how many of those specific PD-L1 break proteins are physically showing up on the surface of the tumor cells. The second is tumor mutational burden, or TMB. This one is entirely different. It's a genetic test that counts the sheer volume of mutations inside the tumor's DNA. The logic here is pretty straightforward. A tumor with a massive amount of genetic mutations looks way more foreign to the immune system, making it much easier for T cells to recognize and attack once the brakes are off.
Next up, Section 4. Challenges in biomarker testing. The standardization roadblock.
Moving from the textbook to the actual lab, we hit three pretty harsh realities. First is spatial heterogeneity. A single tumor can have super high PD-L1 in its core, but zero on its outer edges. So if the biopsy needle hits the wrong spot, you get a false negative. Next is temporal heterogeneity. Tumors evolve. The biomarker profile you test on day 1 might be completely different by day 60, rendering your baseline test useless. And finally, assay standardization. Right now, different labs use different chemical assays and different percentage cutoffs to decide what counts as positive. This lack of standardization is absolutely critical. It's literally the difference between a patient being misclassified and missing out on a life-saving drug or receiving unequal care just depending on which lab processed their sample.
Section 5. The 3PM framework. This is the precision solution.
So, the crucial point is how this framework operates in 3 distinct, continuous steps to solve these exact problems. Step 1 is predictive. We move beyond just checking one single marker like PD-L1, and we use multi-omics to look at the genes, the proteins, and the tumor environment all at once to precisely stratify patients. Step 2 is preventive. Instead of waiting for a scan 6 months down the line, we monitor the patient's blood in real time to catch resistance the exact moment the tumor starts mutating. And step 3 is personalized, using all that data to tailor combinations of drugs, say an ICI mixed with a targeted metabolic therapy, specifically designed for that single patient's completely unique cancer profile.
Finally, Section 6, the future of precision oncology, when tech meets biology.
The tools that are going to power this future are absolutely staggering. We're moving rapidly toward liquid biopsies. Instead of surgically removing tissue, we can take a simple blood draw and detect circulating tumor DNA. This allows us to track the tumor's genetic evolution continuously and totally non-invasively. We're also using spatial transcriptomics to map out the exact location of every single cell in that hostile tumor microenvironment. And to make sense of this astronomical amount of data, we're generating composite multiomics biomarkers, driven entirely by artificial intelligence.
Let's move to and see how this builds, because AI really is the linchpin here. As the research notes, AI-driven models can find these intricate, invisible connections between a patient's clinical history, their genetics, and digital tumor images. A human pathologist, no matter how brilliant they are, simply cannot compute millions of data points simultaneously. AI can, and it allows us to find predictive traits that conventional analysis would completely overlook.
Which leaves us with this final thought. We are transitioning away from a world of trial and error chemotherapy and moving into a highly mapped, dynamically monitored, microscopic war. As AI models become sharper and liquid biopsies become standard practice, are we finally entering an era where your cancer treatment is as entirely unique as your own DNA? Well, the science says yes. The 3PM framework proves that we are closer than ever to truly personalized, precision oncology. Thank you so much for joining me on this explainer, and keep questioning the science.